ठीक है.
Good morning all. So as we are doing this 4-session series, so today is the 4th session and it will be the last session. So here, as I promised earlier, we will be discussing this most probable topics from paper 2 for this year mains. So before that, just a reminder, so classes is going to start from 15th and you may get like this thing discount up to September 12th, right? So with this, we will start this session, right? So I will keep this session very crisp and clear. So here the major focus would be on highlighting the important topics first thing, and for important topics I will be giving like brief discussion also, okay. Brief discussion also, and also for like each topic what are the subtopics, and if at all that question is come in the examination, so what is the structure you can adopt for that. So this aspect will also be covered today, right? So as I told you, the major objective of this today's class is it is for most probable topics of paper 2, right? And which are the major subjects in paper 2? As we know, we have majorly 7 subjects. One is cell biology or else genetics, then it is plant breeding, then seed science and technology, plant physiology, plant protection, the horticulture, and food science and nutrition. So these are the major 7 subjects we have. So I have taken like some of the important topics which is like most probable to be appearing in this examination, and also the same PDF which is available in this video description itself. You can see this video description. There you will get this 2 documents. One is on this most probable topic, and another would be on this GM technology. So that is what is the status of this GM technology, and also like what is the procedure we adopt to develop this GM technology. So these aspects would be there in this PDF. You can download those things. It is available freely in this video description, right? So with this, then we'll start with this first subject, that is cell biology and genetics, right? In this cell biology and genetics, see, first and foremost, I told you like many a times what are the important aspects in this. See, first how to like decide which is the important aspect here. First, let's say for example, if I want to test your knowledge on any subject, first I want to know whether you know the basics of it or not. If you have understood what really the fundamentals of that subject is. So if I say, if I want to like test your knowledge on this genetics, which is the fundamental knowledge I should be testing for you? Obviously it is cell biology, or else the how cell functions, what is the structure of the cell, what is the difference between prokaryotic and eukaryotic cell, what are the different cell organelles, what are their functions, and like the regulation of the cell cycle, the stages of the cell cycle, everything is going to be important. Because if you understand this only, then you will be understanding what are chromosomal aberrations, what what is heredity, what is this genetic code, what is photosynthesis for protein synthesis for everything. So the cell biology is the basic. So that's why. So if you have observed like questions from this genetics, at least one question will be there from cell biology in one way or the another every year. So in other words, cell biology is going to fetch you like at least one question which will be worth of like 10 to 15 marks for sure, right? Okay, so that's why that the important topics which I have predicted for this year mains, one is cell cycle, that is, and also like cell cycle regulation, you know, like cycling kinases, etc. These are involved. So which will regulate how much of this cell cycle should happen. If at all, let's say for example, uncontrolled cell division is there, then that condition is called as cancer. Yes. So that's why. So we should have this regulated way of cell cycle. So what is the procedure, or else like what is the mechanism involved in this regulation of cell cycle? That is also one of the important aspect. And mitosis, the question on mitosis in like last few years it has not appeared. So this year there is high probability that it could be asked, right? Next one is semi-conservative method of DNA replication, right? Here there are actually many types of DNA replication. For example, conservative method is there, for example, dispersive method is there, and also semi-conservative method is there. So what is the difference between these three, and also like what is the procedure, or is like mechanism which is happening in this semi-conservative method of DNA replication? That is one thing, along with what is that, the diagram. So that you should be knowing. Clear? Next one is law of segregation. See, without Mendel's law, so understanding genetics is tough, and also like it has given like new facelift to like understanding of genetics. So that's why, so Mendel's law is again going to be very important. So in that law of segregation, so that would be the important law for this year, right? Next one is chromosomal aberrations. So here either structural or else like numerical could be asked, but my feeling is that this year the structural aspect could be asked, that is structural chromosomal aberrations could be asked this year, right? Next one is differences between linkage and crossing over. See, these two are very important because in this way, like in, see, for example, in crossing over, the natural variation is created, correct? And also in the breeding, we exploit this variation. So that's why the knowledge of crossing over is important. But if you take this linkage, so linkage is one of the hindrance, or else let's say, for example, one of the, what is the problem in this characteristic transformation from one organism to another organism, or else from one plant to another plant, correct? So that's why these two concepts are important. Next one is self-incompatibility. So here you have to understand what is the concept of it, what are the mechanisms. In this mechanism, when I say you have to understand both like at cell level and also at gene level. So these two things are important here. Next one is sex-linked inheritance. So if you have observed the syllabus, in the last line of this syllabus, we have something called as sex-linked, sex-limited characteristics. So there sex-linked characteristics could be a potential question for this year mains, right? Now what I will do is like as I explained yesterday also, at least for genetics and breeding, I will be explaining in little bit detail for these concepts, and for the rest of the things I will be explaining in brief, right? Okay, so with this we will start this first concept, that is cell cycle. Okay, so cell cycle is very very important because it is a programmed thing which will be repeating itself. So first thing what we have to understand from cell cycle is like the procedure, what are the different stages of it. So start from this GO phase, right, that is sort of small resting phase. After that go for this G1 phase where exactly the cell growth will happen. After that in S phase, that is where the synthesis, that is synthesis of DNA or as genetic material, that will happen in this. So in G2 actually small amount of synthesis of DNA will happen, but mostly it is a stage of this mitosis. And then in this cell division will happen. So in this cell division, obviously we know this phases, prophase, metaphase, anaphase and telophase. So these things will happen. After that cytokinesis, that is division, will happen, and then again this cycle start to repeat. But important aspect here is that if at all the question like explain this cell cycle, it has been asked in this examination, first you have to start with this definition. So say what is cell division. After that you have to give this diagram and explain each stage in very brief form, right? And after that you have to, like, even though it is not mentioned in the examination or it's like in the question, let's say the regulation aspect is not asked in the examination, even then you have to give this regulation aspect. Because so if you can see here, I have taken like 4 of this regulation methods, but essentially there are n number of things. So one, as I told you, like cyclins and cyclin dependent kinases. So depending on the concentration, how much of this like chemicals is produced in this cell, so the cell cycle will also be determined, correct? So same thing with respect to this checkpoint proteins. So these proteins, again the concentration is very important. Okay, oncogenes. So oncogenes, you would have heard, these are related to this cancer. So in cancer, what is the stage? As I told you, it is uncontrolled cell division. So here also, like the pace of cell division will be increased if at all this oncogenes will be present. So regulation or is like presence of this oncogenes is also one way of regulation of cell cycle. Next one is apoptosis. So this everybody would be knowing. So apoptosis is also called as programmed cell death, right? So this is like where if at all the cell cannot be repaired, it is beyond repair, then cell itself will it is going to like kill itself. So that is called as programmed cell death, right? So this apoptosis, cell senescence, these are the basic aspects you should be knowing. Plus whenever the question is there, as I told you, if all the question is like this, explain cell cycle or else define cell cycle, explain the phases in cell cycle, then explain those things, but in the end part definitely give this regulation aspect also, which will be acting as like like completeness to your answer. Clear? So this is one aspect, that is cell cycle and regulation of cell cycle. The next one is mitosis. So we know like mitosis and meiosis are very important ones. So in mitosis, how many like daughter cells will be created? That is 2 daughter cells will be coming, correct? Yes. So explain like each phase, that is interphase, starting from there, prophase, metaphase, anaphase and telophase. See, here the problem is not with respect to sorry. Here the problem is not with respect to content. Here the problem is that like student feel like whatever he know, he has to write everything in the answer. See, these are the questions where, like, if at all it is very repetitive and for sure it is going to appear in the examination, you should be smart enough to write those sort of details which others might be missing, and at the same time you should not be compensating on the looks, that is aesthetics of the answer, that is presentation and structure should not be compromised. So when you are like, let's say for example, this sort of like overall one diagram could be provided. After that, when you are explaining like each stage, like let's say for example, if you are explaining this anaphase, so what are the major events which is happening? And don't forget to give this, like what was the thing happened in the last stage and how it is being continued in this present stage. So this continuity should be there in the points, plus what is the keyword, that is which is the most important activity which is happening in this phase, right? So that you have to mention it. Yeah, so that is the most important aspect of this question. So here, as I told you, it is not about, like, it is not difficult about the content, it is about the presentation. Like as of now, since like last week is there, what I would recommend is that don't worry about the content, worry about the presentation and structuring. So whenever there is time, rather than like opening the books and by-hearting the things, what I would recommend is that open the books, but essentially not like without reading from the books, close your eyes and try to like remember the things. So say for yourself, okay, after this this will come, after this will come. If at all that flow is not coming, then go to that particular page which is a particular point you forgot, and think of it whether you forgot because like you didn't know the concept, or else you forgot because there was this lack of connectivity between the points. So think of that and do this interlinkages, then that would be better way of remembering the things, right? So do, like see, last 7 days it is mainly your orientation should be like this. First thing, I would remain calm, so no matter what, so whatever is the like condition, okay, let me do this. So I will take like 5 minutes here in between, so I will tell you like what should be your approach or else like mindset in this last 7 days, right? Okay. So last 7 days, obviously whatever the like reading activity should have been done, so that would have been done now. So don't worry about like reading extra, or somebody is coming and saying that, okay, you read this, so this is like compilation of this thing, that thing, doesn't matter. See, at the end of the day, there is no guarantee that that question will be coming, right? So that is one important thing. Second thing is that even if you like try to read that in this pressurized environment, obviously that will not be going to your brain. So rather than worrying about like what is the thing which you have not done, so forget about that. So focus on what is the things which you have already done and try to bring quality in those aspects, right? So that is one important thing. See, I have seen like students having this habit of like going to temple on the examination day. So that is not bad. So I am not like atheist or something, but what I am trying to say is that if at all you are going to do that on the examination day, try to do it by now itself. Like at least for the 7 days, if you are doing that, what is the psychological advantage is that your brain will be tuned in such a way that, okay, tomorrow I will be going to the temple, after that I will be writing this, after that I will be revising this. So that tuning is important. If you are going on a particular day, that is on the examination day, to this temple, what will happen is that your brain becomes very much conscious. So it will start focusing on minute details which will be happening on that particular day. So this minute details, or like being aware of like every small details in a day, that will not allow you to have common sense. So on this examination day, the more important aspect is common sense and presence of mind. So that you will be losing if you are doing like specific activities and with special emphasis on the examination day, right? So do avoid that, or else if you want to do that, do it from now itself, right? So that is the second thing I wanted to say. Third thing, with respect to revision, so it is, as I said, though rather than like looking for new things, so whatever the things you have already done, in that cost to benefit ratio. So as I given this high yielding topics list, so do like keep this list, and accordingly you just remember the things. Okay, this is I have studied. If what are some new concept is there, let's say for example that systematic weight intensification, if that is a new concept for you, okay, do learn about that because that is in the news. But rather than focusing on the unknown things, it is more important that whatever the things you already know, so do like memorize them. Memorize in the sense, it is like what is that visualized memory, that is picture form of memory, rather than just by hearting the things. So this is the third important aspect. Fourth important aspect, I am sure, like pretty much sure that everybody would have like stick to like one pen, and in that pen only you would have practiced. So on this examination day, if you are writing in this gel pen and all, so ensure that at least like adequate amount of refills is there, right? And do take them to your examination and all, and don't get in quarrels with this, invigilator or else like the coordinators who would be there. See, if at all worst case scenario is that bench would not be good, the fan would be making sounds, or else let's say for example in outside, or else like outside your school there might be some function, they would be like screaming, etc., etc. So be mentally prepared for all this sort of like uncontrollable things. So rather than worrying about this uncontrollable things in the examination hall, it is much better to like be prepared from beginning itself for those uncontrolled situations and focus on yourself. Clear? Now, with respect to pen, if at all it is making this, like especially gel pen, if you are starting to write more and more, it will start to like leak little bit more, right? The ink will be there in particular alphabet little bit more. So for that, what you can do is that you can take this small cloth, so you can keep that next to your palm, so I mean below your palm, and you can write on that. So once there is like excess of this ink coming out, then you can use that cloth, clean that, and again start writing. So if possible, if you are not like practice with this gel pen, if you have this like a V7 pilot pen or something like that, so that would be better. So there the ink would be the problem. So rather than changing ink cartridges, etc., don't waste the time. So buy like at least 2 or 3 extra pens and go with to the examination now. And fourth most important thing is that don't go with any sort of expectations. So literally no expectations should be there, and whatever the things which you have learnt in the like preparation phases, that is in the preparation phases. Don't think that whatever you read, it will come in the examination. So your strategy should be such that, okay, whatever they may ask, so at least I will be knowing something about that. So I can read the question, understand the question, identify the subparts, and address the demand of the question. So this should be your approach rather than having that biasness. Okay, this is the most important topic, so question will be coming from this. So that will be is like going to create biasness. And if at all that question is not there, then you will be panicking. So especially the people who are writing mains for the first time, for them this would be the problem. But essentially don't panic in the examination hall. So initially, see, for example, even the first ranker would be having that nervousness in the beginning of like 5 to 10 minutes. So first start writing this 1 or 2 questions, then that nerves will be calmed down. After that the flow will come. So that's why in the beginning phases, don't harm yourself. Clear? Okay. Next important thing is that finalize your strategy in this 7 days. That is to say, when I take up the question, how much time I would be spending on like reading the questions, or else somebody will be having this habit that they will go from question 1 to question 20 in one go. Without reading the questions, they will be okay with their attitude. They will be like, if at all an unknown question is there, it is fine, they will be moving to the next one. So that would be there. For you, if at all, it is like spending little bit time in the beginning, like identifying which is your easy questions, whether to solve like 15 markers first, 10 markers must, or let's say for example in optional it is 20 markers or else 15 markers, whatever is your strategy. So finalize on that and do implement the strategy. See, problem with the students, especially people who write mains, is that it is not the problem of planning, it is a problem of implementation. They would have had like very good strategy. Okay, I will do this, I will do this, I will do this. In the examination, when they see like unknown questions, or else like suddenly there is some environmental crisis, that is in the examination hall, the environment is not like conducive for you, then you will be panicking, then whatever the strategy you had, you will be like deviating from that, then the implementation problems is there. I'm not saying that if you implement your exact strategy, you will be getting first rank. But what is that going to give for you is that satisfaction. See, when you come out of this examination, at least you will have that satisfaction. Okay, whatever I had thought of like implementing, I have implemented. So there won't be like regrets. If not, you will be always regretting the things like, okay, I could have done that. I had already actually thought like that. I should have done that. So that will not be the case. Are you getting? So that's why stick to your like strategies which is working for you. So in the nth minute, if at all somebody is coming and saying that this is the strategy, etc., don't do that. So that's why in the like end phases, I won't be saying this strategy. Okay, first attend this 20 markers, 5 markers. No. So what I have to do is that whatever the strategy you have figured it out for yourself, do stick to that. Are you getting? Yes. So these are the things I wanted to tell for like people who are appearing the mains. See, at this point of time, rather than like content and all, I believe that it is like psychological and mental preparation. That is the crucial aspect of this last 1 week. So focus on that, please, and like don't waste this attempt. See, especially if you are writing mains, means like lakhs of people will write the prelims. So only lucky few will be allowed to write the mains. In that again the filtration will happen, only lucky few will be going to the interview list. So that's why make good use of the opportunity which is thrown at you by the life. Clear? Right. Okay. So with those things, that is like good enough. So now we'll be focusing for this next part. Okay, so I hope this mitosis is clear. Okay, next one is this semi-conservative method of DNA replication. See, as I told you, like there are n number of types, but majorly like three types is there, or three ways of this DNA replication. One is conservative, second one is dispersive, and third one is semi-conservative. So in this conservative method, what is happening is that, if you can observe here, see, this is the original DNA, and this is the first replication which is going to happen. So here two different types of like original and new type of like DNA would be created. So here the original type is conserved, so that's why it is called as conserved type of replication. If you take this dispersive, so you can see the mixing here, half this, half that. So here what is happening is that the replication, it will happen in the pieces. That is the original one, it will be broken down into different pieces, and then like the joining will happen. So that's why the recombination, once that is done, and the replicated one, it will be mix of both the things. So that's why it is called as dispersive. So disperse means what? It is it is separated. So that is the meaning of this dispersive. Next one is semi-conservative. So in this semi-conservative, so one strand will be of this new DNA and one strand will be of this old DNA. Now the old DNA is also conserved, but at the same time new DNA is also conserved, created. So that's why it is going to be called as semi-conservative method of DNA replication, right? And essentially you should be knowing another two important words here. One is autocatalytic function and heterocatalytic function. So autocatalytic function, it is pretty simple. So auto means what? Self. So here DNA is like directing itself to create its own DNA, or else like replicate its own DNA. So that's why it is called as autocatalytic function. So heterocatalytic function, because we know that utilizing this DNA, we have this genetic code, and that genetic code is pretty much important for protein synthesis. So that's why heterocatalytic function, it says that it is going to give directions for the synthesis of RNA and protein synthesis. So these two are the important functions. So when question is there, like explain this semi-conservative method of DNA replication. So one way of starting is that, okay, you give this, who has given this evidence for this semi-conservative method of DNA replication. So even I have given here, it is Michelson and Stahl. So these are the people who have like proved to the world that the replication method is semi-conservative. But at the same time, another way of starting this answer would be, like you can start with this definition of DNA, right? After that you can give these two functions. After that you can explain about this model, like semi-conservative DNA model. Clear? Up to here everybody is clear, right? So this is the diagram which you can write, or else let's say for example, even simplified version of like diagrams would be there. You can write any one sort of diagram, that would be good enough, right? So like here the main part in the answer is the enzymes involved and also the phases. For example, here it should be like the leading strand and this lagging strand. So how this separation will happen. So then how this new DNA would be formed, new strand will be formed. So this DNA polymerase enzyme. So these are the main important details you should be providing here. Are you getting? So main focus would be on like the technical terms used here. Second important thing is that the flow of like proceedings. That is, once this unwinding is done, then next what is happening. So that flow should be there rather than like explaining in a haphazard way. Are you getting? So here again, as I told you, like content is not a problem. See, especially if you take like paper 2, here the content is not the problem. Here the main challenge would be to present well and structure well. Clear? I've been telling you from the first session itself. See, focus should be on like good presentation and structure rather than like dumping off more and more of knowledge. Are you getting? Right? So this is about semi-conservative method of DNA replication. Next one, see Mendel's law. Again, this is one of the most important topic which is like repeated n number of times. So obviously, like usually two types of laws will be asked in the examination: law of segregation or as law of independent assortment. right? So law of segregation, it is pretty simple. So but if at all law of independent assortment is given, then you have to like take different characteristics and then you have to like put this checkerboard and all. So that's why, so this is like easy one, and if at all this is going to be asked, either it will be asked for 10 markers or 15 markers, right? So when this question is asked, start with this rediscovery of Mendel laws, so which happened in 1900s. After that you say that, so the thesis statement, that is what is this meaning of law of segregation. So they basically say that if at all gametes is formed, so it is not going to be mixed, so that in the next generation also the gametes would be separate. There is no intermixing between these gametes. So that is what in the simplest understanding about this law of segregation. And that's why purity would be maintained in the further generations also. Just establish this with an example of character. You can start like give this example of P, which like Mendel himself had done, or else you can take some other concept or some other example and then also you can present the same thing. Clear? So here the main important thing is that one is this checkerboard and also giving that purity of gametes is maintained. Clear? Okay. Next one is chromosomal aberrations, and again this is like one of the most repeated topics in this genetics. So alternate years, at least one question either on like numerical or else on the structural it will be asked. You know, so like in the like present trend which is going on, so I believe that this year it would be of the structural chromosomal aberrations, right? Because structural have not been asked like for some years. I think that this year it might be the case for structural chromosomal aberrations. Now obviously the numericals I have given here, so better even if at all it is asked for this, you know, like structural or numerical, first give this flow chart, right? Like first say this what is like chromosomal aberrations, and then briefly mention why it is going to happen. If you can see here, yeah, so if you can see here, so these are the reasons I have given. So one is like spontaneous errors in this DNA replication. So second one could be like radiation exposure, like gamma rays, X-rays, that sort of thing. Next one is transposons. It is also called as jumping genes. And again this question has once appeared in the examination. So jumping genes or as like transposons usually are dubbed as like junk DNA material. Is that true? But essentially in most cases it might be true, but it is also having like utility. So just for your curiosity purpose you can think about this transposons also. Plus another important, especially for this numerical ones, is like non-distinction of the chromosomes. If you take this, okay, if you take this you know like cell cycle, so here especially when they have like you know arranged in this metaplate, after that it is going to move towards these poles. So while moving, what will happen is that a fragment of this DNA, either it will be lost or else addition will be coming. So that's why in numerical chromosomal aberrations we have is like addition and deletion, it will happen. So like triploid condition, tetraploid, so this is about numerical ones. So if we take the structural ones, so here what is happening, either like a part is deleted, then duplication, so extra will be coming, then inversion and then translocation. So these are the major types, and here when you are explaining ensure that you are going to give this reasons, cytological reasons, and then impacts, and then you move to the another one. So and also like if possible, whatever you are trying to convey in the words form, you can give that in a small diagrammatic representation also. Because if you say inversion, it is pretty easy to show, correct? So that sort of like diagrams you can inculcate in this answer, that would be better. Clear? Now let's say for example for all these four things, it has been asked and it is just for 15 marks. So 15 marks means there will be like 3 pages, correct? So if at all it is just for 3 pages, what you can do is that, so rather than like you know compensating on the points, what you can do is that presentation or else like present this flow chart in the beginning and also give the like subtypes. After that explain at least like 3 important types under each one in detail, right? So that also is one way of presentation. And second way is that, so present the same thing, that is to say explain all the things, but the points could be like prioritize the points, that is most important points only will be provided and then you can complete the answer. So that is also one way, that is prioritization of the points. So when there is like a good amount of content, prioritize and make a list. Okay, if I have 10 points, in this 10 points which is the top most 3 important ones, and try to write those 3 important points in this thing. Clear? Right? Yeah. So this is about like you know cell biology or else genetics. See, important thing is that first and foremost thing, you have to remove that fear in your mind, thinking that genetics and breeding, it is going to be tough. In essentiality, actually it is not tough. So the toughness is basically you are feeling because of like, the things are being by-hearted, that is one. Second thing is that you have not done this PYQ analysis. If you have done like PYQ analysis, then what are the topics which I have just mentioned, you would have been thorough, right? So up to here it is clear, right? So next one is plant breeding. So in this plant breeding, I've been telling many a times, see basically the idea of this plant breeding itself is simple thing. First thing, create this variation, then transfer that variation into the plant which we want. After that, mass multiply it. When you are doing this mass multiplication, it doesn't mean that everything or else every plant is going to have this variation. So selection, so that is first is creation of variation. So for that we have X number of methods. For example, let's say mutation, that is one way of creation of this variation. So after that, you have to do this, mass multiplication. During that, we are going to like have this selection procedures. For example, for example, this mass selection is there. For example, this clonal selection is there. So this sort of like selections would be followed, and then we are going to pick up this top performing this varieties. After that, those varieties will be multi-location trials. After that, we are going to have this seed multiplication. Then it will be given to the farmers. Correct. So this is the overall understanding of this plant breeding. But essentially considering all those things plus the PYQ trends, I believe these are the important topics for this year mains. Right. One is backcross method, and again this backcross, for example, when it is used, why it is used, and the procedure for this. So this is going to be important in backcross method. Next one is inbreeding depression. So this is very important. See, the important aspect you should be having clarity is that cross pollination and self pollination is there, but this cross pollination actually it is more susceptible to this inbreeding depression rather than the self pollination. So you have to understand why that is going to happen. See, because in this cross pollination, basically it is developed itself to have this like different pollination method, that is basically like cross pollination. If at all a little amount of this selfing it is going to happen, it is going to create this inbreeding depression. So that's why susceptibility is more, the degree of tolerance is less. But in this self pollination, if you take, there the degree of tolerance for this selfing it is more. So that's why inbreeding depression is observed more in cross pollinated crops rather than in self pollinated crops. And again, this is again one of the PYQ which is asked many years back, but essentially that is again one of the potential question. So if they want to check your fundamentals, they will be doing that. Or else if they want to like even make it much more easier or else like fundamental for you, then they may be asking like modes of reproduction. So like 3 types of pollination is possible, or else like pollination, that is cross pollination, self pollination, and often cross pollination. So in these things, which are the morphological modifications which is happening in the plant to promote one type of like pollination. So those can also be asked for you in the examination, right? So that is inbreeding depression plus self and cross pollination crops. Next one is marker assisted selection. This is not asked like in last 2-3 years, but essentially this is going to be very, very important topic. But essentially this has been asked like n number of times. Here again, like I've been telling you in the genetics, here also it is the same story. Here content is not the difficult one. So I will give you the structure how you have to present it when that topic is going to come. But essentially here, as I told you, important focus should be on like giving this conceptual clarity plus good amount of presentation. Clear? So I will give you the structure, don't worry. Next one is this thing, GCA, that is general combining ability. If I want to have a breeding program, so what is the compatibility between 2 parents? So that is like the understanding which we want. So that is called as GCA. For general characteristics, it is general combining ability. So for this, of course, the selection procedure we have is recurrent selection. So GCA for recurrent selection for GCA, that is like a exact flowchart itself is given in this BD Singh textbook for breeding. So you can read from there. And pretty simple, actually it is very simple. So you just take this recessive parent and then do this crossing again and again. So that is the basic understanding of this. So I will show you this diagram also, right? Next one is GM technology, and again this is going to be important this year because like n number of articles was there in the newspaper, first thing. Second thing is that this GM crops, especially GM mustard, so that was in news. So, and again, like this is also part of the syllabus itself. If you have observed, so GM crops, that is genetic modification, so that itself is part in the syllabus. So that's why this is again one of the potential question for this year mains. Clear? Now, I will take like few of the important topics here. I will discuss that in brief, and rest of the things I will, tell you the structure, what you can follow, right? Okay, so this is for that back cross, right? So if you can observe here, so here I have given like why we are going to use this back cross, or else when we are going to use this back cross. So first one is for intervarietal transfer of this inherited characteristics. That is, you have like two varieties. One variety actually it is having like very good agronomical characteristics, but it is susceptible to this pest and diseases. So another variety is there which is having like a very good resistance but poor agronomical characteristics. Now if you want to like transfer the ideal characteristics between these two, then you are going for this back cross method. And also this one is important, third one, that is interspecific transfer of this simply inherited characteristics. For example, interspecific means what? Within the species. Within the species, if you want to transfer these genes, so then that is one of the thing. Correct? Here it is like, for example, transfer of disease resistance from related to this cultivated species. That is, you know, this Gossipum hirsutum, it is for cotton. So here resistance of this black arm disease in the cotton from this wild tetraploid species, that is incorporated. So within this species, if at all there is one resistant character, that can be transmitted to this cultivated character. So these are the like cases in which we are going for back cross method. So ideally for the structure of this answer, what you have to do is that okay. Right. So here I was saying for black, okay, back cross. So if at all question is asking, okay, question is asking like explain back cross, and usually it will be asked for disease or pest resistance breeding. So in this case, first start with this definition only, that would be better. Start with definition of back cross. So here important one is give proper definition, correct? After that you can give this example of famous varieties developed using back cross. After that you can go for procedure. Sorry, before procedure what you can do is that cases or else situations in which we can go for back cross. So these things actually, whatever the things I am saying, it is already present in this ICR notes. So if you go to this ICR e-notes, then you will be finding this content there itself, right? So give these cases. After that you can go for this procedure. So in procedure also, don't spend your time like explaining in like passages. It is much better to have this flow chart. In flow chart, like it is easy. For example, this thing, first year, second year, and then 4th to 5th year, then 6th to 8th year, then 9th to 10th year. So this sort of classification you do. So major events which will be happening here and major outcomes which is happening in each stage. If you just present that much, it is good enough. Clear? Because that is much easy for even evaluator to understand, and also it will be easy for you to represent the content. So that's why it is much better way of presentation of the content. Clear? So that is for this back cross. So same procedure is being given here, right? So if you can see here, it is a resistant cultivar, but this is an adapted variety. So now it is going to be crossed in this way. Clear? Right. Next one we'll take is, okay, MAS, that is marker assisted selection. Okay. So first we will understand this, what is molecular marker. Then I will give you the structure. What is the structure you can follow in this answer writing for this molecular markers, right? Right. See, as the word itself is saying, it is molecular assisted marker assisted selection. So here markers could be of these three ways. One is morphological markers, next one is biochemical markers, and third one is molecular markers. So these are the three types of markers we have. So morphological, it is pretty simple, like anyone can like identify it. Let's say for example plant height, let's say for example leaf size, let's say for example the number of flowers in each plant. So these are the morphological characters which an human eye can easily see, and we can use that as a marker for identification of better varieties. So that is this morphological markers. Next one is this biochemical markers. In this biochemical markers, like for example presence of enzymes, like isoenzymes, if at all the quantity, how much it is present, and also if at all presence or absence of that, that is also important. So those will be used in biochemical markers. So next one is this molecular markers. So within which we have this PCR based and non-PCR based. So within this non-PCR based, we have this RFLP, that is the random fragment length polymorphism, right? So that is one. And within this PCR, we have again like n number of times. So some of them I have like abbreviated, abbreviated form is given here, right? You can see for yourself. But essentially in this molecular marker, what is happening is that now let's say if I want to transfer this disease resistance or else pest resistance or else like good yield characteristics, which is governed by a gene only, because DNA is the main vehicle of heredity, and it is the one which will be causing this transfer of the characteristics from one generation to another generation. Now what we are going to do in this molecular markers is that, for example, for each individual characteristic there will be one gene. Now for this gene there could be this molecular markers. So this molecular markers will be tagging that and we will be running this PCR. So PCR is polymerase chain reaction, right? So that equipment will be there. You just pass that the material which you have taken, so that will be running in that the gel electrophoresis, and then the bands will be generated. So depending on these bands and the length, and also like let's say for example the characteristics of this band will be taken into consideration, then we will say that okay, if at all this plant is having this resistance, then that band will be the characteristics. So depending on that we are going to take this band, plant. So after that we can adopt like any other procedure. For example, we can go for like normal multiplication process, then that will be different. But for identification, but for selection, we are using this molecular markers. Up to here it is clear? Yes? Yeah, same thing is given here if you can see. So this one, 2 sample sizes are there. So then after that we are going to self-fit, then a population is generated. In this we are going to take this DNA from each individual, and then we are going to do this polymorphic primers. After that this gel electrophoresis, as I told you, it will be run. So this is how the bands will be formed. So depending on this, let's say for example the band is present like this, then we have this marker, we have this resistance, then that will be selected for future generations. So this is a pretty simple concept. Now I will tell you this answer or the structure, how can you do for it. Now, okay. Okay. So MAS, marker assisted selection, right? Structure. If at all the question is asking like explain MAS, that is marker assisted selection, and we are considering this for 15 or 20 marks, considering 3 or 4 pages would be there. No. Right. So considering first scenario, that is 3 pages, what you can do is that, so question is on explain MAS and procedure, etc., etc. Now first start with this definition of molecular marker. So this is one way of introduction. Second way of introduction would be the necessity of molecular markers. So here you can basically say this disadvantage of morphological markers. because even though if at all morphologically it is present, there is no guarantee that it will be present in the genotype of it. So that's why we have to be sure. So this thing you can say for introduction, clear? After that introduction, you can start giving this flow chart, where you can give this molecular markers, then this classification which I just told you, morphological, biochemical, molecular. Within this subtypes you just give it, and then after that what you can do is that you can take a parentage, right? So any one parentage of a variety you just take it. So 2 parents would be here. After that, as I told you in this flow chart, so same sort of like presentation could be given here. After that, here you can give this advantages and disadvantages. So problem with students is that they will focus on this. They will focus more on this, and they will start explaining all this RFLP, PCR, etc. in the answer. So that is not essentially needed. See, technically this is a breeding question rather than a biotechnological question. If at all it was a biotechnology question, explaining those things would make sense. But here the main focus is on breeding procedure. So that's why emphasis should be on this breeding procedure. So explain this procedure adequately. After give this advantages and disadvantages, that would be good enough. Clear? Now let's say for example 20 marks and 4 pages is there. So if that is the case, then you can explain at least 1 or 2 molecular markers in brief, right? So that would be good enough. Clear? So see, you have to be smart enough just by looking at number of pages, number of marks, and what are the subparts of the question. You have to like have that feeling of quantifying the things. Okay, for this I will be writing this much space, or else this much points is good enough for this. So that sort of like feeling you should be getting. So that is the much better way. Clear? Okay. So this is molecular markers, right? So next one we have is this GM technology. Okay, before we discuss this, so recently this GM mustard was in use, yes? So can anyone tell me, okay, if I say this, okay, let me ask like this. So observe the statement carefully. So as of now, only GM mustard and GM cotton have been approved for commercial cultivation in India. Is that true? Anyone? Even online people, they can like say their answers. When I say GM mustard and GM cotton are the only 2 crops approved by the government for commercial cultivation. Is that true? Essentially, that is false, because actually there are 2 words. One is called as commercial release and second one is called as environmental release. So in this commercial release, what is happening is that we are releasing to the people, that is for the farmers, to commercially cultivate the crop. So in this environmental release, what is happening is that we are releasing this in different environments, and we are going to like have idea like in this environment is it going to perform better, in this environment is it like yielding better. So in this way we are going to do this multi-location trials, multi-environment trials. So that is called as environmental release. So as of now, when this GM mustard was in use, essentially GEAC, that is Genetic Engineering Appraisal Committee. So under this Genetic Engineering Appraisal Committee, they had given this approval for environmental release but not for commercial release. So as of now in India, we have just 1 crop, that is BT cotton, for commercial cultivation. That important aspect you should be remembering. Second important one, this EPA Act, Environmental Protection Act. Under this, the penalty will be like imposed if at all like a crop which is not cleared by GSE will be commercially cultivated. Are you getting? A penalty of up to like 1 lakh rupees, sorry, up to 1 lakh rupees, or else like 5 years jail would be there for that. Clear? So these sort of like basic things, if you remember, it will be helpful for you to clear prelims, and also like that will be value addition in your answers, right? Now, so this GM crops, it could be like this. For example, explain the status of this GM crops in India. 1. Second thing is that what are the tools utilized for this GM technology. Third, what are the potential risk and also like solutions for this GM crops. So these are the aspects you should be looking for under this GM technology. So first one, I will just let you know this brief procedure, how we are going to do this GM technology, and in this procedure only few of the tools will be mentioned. Apart from that, you have to like look for yourself. Clear? So if you can see here, first thing is cutting out the gene. That is, for example, you would have heard about this, golden rice. Yes. So golden rice here, the genes from this daffodil, the flower, from that it was taken. So here also, like first the source will be identified, in that using this molecular scissors, right? So utilizing that, we are going for this like cutting of the DNA. So essentially here another technology you should be knowing, that is CRISPR-Cas9, right? So that again it is in use. So that has been asked in GS3 environment and also in this prelims. So that could be a potential question in our optional also. But essentially, first thing is molecular scissors. It is like a small part of this GM technology, right? Utilizing that molecular scissors, you're going to like get this portion of DNA which you want. After that, it has to be like transferred to this vector DNA. So for this vector DNA, like something called as plasmid, so this will be used. So here in this plasmid, so we are going to take this portion of DNA which we have taken, and then we are going to put that into this vector. Then essentially that is called as rDNA, because it is recombinant DNA. So rDNA or as recombinant DNA would be there. Now after that, we have to like mass multiply it. So for that, we are going to incorporate that into this bacterium. For example, agrobacterium, or for example some other bacteria could be used, but essentially usually agrobacterium will be used. So this agrobacterium will be used to put this rDNA into that. After that, so something called as this bullets is there. See, essentially in the gun you use this gunpowder, right? So here what we are doing is that, so in this this thing, what is that? Teflon bullet. Teflon is a material. So on that we are going to load this DNA, that is basically vector. So after that, this is the gene gun, how it looks like. Utilizing this, you just incorporate that into this plant cell. So because it is going at high velocity, it will be directly entering into the plant cell. So these are like basically like important thing till the incorporation. After that, after this gene is entering into the plant cell, it will get to like incorporate into the plant DNA. After that, those are called as like GM crops. Now once that is done, then we will go for this mass multiplication. So in this mass multiplication, we will get to know like whether it is a success or failure, whether this intended target, that is intended gene, is transferred or not. Then if at all that is the success case, then as I told you, like environmental release will be there. After that, it will be given for commercial release. Okay, just a second, I think few doubts are here. Yes, offline open session is there. And PDF, it will be in this description itself. Right, so in this video description, the PDF would be there. And also as of now, the offline session is going on. So if you want, you can come also. Sorry. So this is the GM technology in brief. So for current status of this GM crops, what you have to do is that one best source is PIB. So Press Information Bureau, you just go and look for this GM technology and what is its use in India. So that would be much better, correct? If not, I think I have shared this document in the video description itself. There also, like, I have given the procedure plus the what is that status, not just in India, in the world itself. What is the status of this GM technology that I have provided there. So do read from those things. Clear? Right. Then that is for this plant breeding. So next one is seed science. So seed science, again, like, see, for example, if you understand this genetics and breeding, then understanding physiology and seed science is easy. Because essentially, see, whatever the things you are going to do in this, understanding this genetics, that will be applied in this plant breeding. So after this plant breeding, it has to be commercially made available to the people. So for that, we are going to have this seed science. So that's why three subjects are interlinked. So when you are reading, also think in that way only. Clear? Okay. So under seed science and technology, these are the important parts. So one is principles of seed production. One is genetic and agronomical principles of seed production, where what is this isolation distance, what is how to maintain this genetic purity, all those aspects will be covered. Next one is, okay, the spelling mistake is there, it is hybrid. So hybrid seed production in this bajra, and not just bajra, actually even in rice is there, maize is there, but essentially Like since this has been asked once, so you can look for like rice also. That is again one of the important crop. This is like a very less asked question, but a potential question for this year mains. That is role of this public-private partnership in seed production and marketing in India. Right. Last one is IPR regime, which is PPV-FRA. That is Protection of Plant Varieties and Farmers' Right Act. So like here I will be like briefly explaining few of these aspects, and rest of the things I will be saying in the form of structure, right? Okay. So first one we will be looking is genetic principles. Genetic and agronomical principles. See, basically genetic principles means what? We have to ensure there is like genetic purity in whatever the seeds you are going to produce. For that, what are the measures you have? See, for example, if you take this, so these are the like types of deterioration or is the way in which it is going to be impacted, correct? So here if you take developmental variations, that is like grown in different environment, soil, climate, that is also going to like compromise on this genetical quality. So for that, growing seeds in their adaptation or else in their growing season, that will like reduce the genetic manipulation, or it's like genetic what I can say is, okay, so basically the genetic composition, it will remain intact if at all it is grown in its own growing season or else it is grown in its own environment, correct? So that is one way. Next one is mechanical mixtures. So at the time of sowing, like when the time of harvesting, the extra material could be coming into your mix, so that has to be removed periodically. Mutations, so depending on the like nature or else like environment in which you are going to preserve, if at all like harmful UV rays or else like X-rays or else even gamma rays it is exposed, then obviously like there will be mutations. So better one is that remove this mutant variety, and those sort of like measures you can take. And again this is like clearly given in this Tamil Nadu notes itself. So just ensure that this fundamental question, that is principle of like seed quality, that is genetic principles and agronomic principles. In this agronomical principles, main thing is isolation distance, and this isolation distance itself could be a potential 10 markers or 15 marker questions. So again do ensure that you are going to read a little bit about this isolation distance. Clear? So this is the principle. Next we will look for is PPP, that is public private partnership, right? Here what I have done is that I have taken this overall like scenario, like how it started. If you take like entire seed supply chain in India, so how it started and now how it is progressing. So this is what it is trying to convey. So if you take like early 1960s, when there was this green revolution, obviously hybrid production was very important. So that's why the domination is from public sector. So NSC, that is National Seed Corporation, that was established, and majority of this seed production and marketing, it was directly done by the government itself. So that's why the domination is from government sector. After that, during before this economic reforms, that is LPG reforms. So what we had is that the private domain was trying to enter into this public domain or else into this public market. So that was happening, but essentially it was like still being like done by the public enterprise itself, like majority was under public itself. After this LPG reforms, that is from 1990s, that is mid 1990s till like early 2000s, what was happening is that this IPR regime, right, intellectual property rights and the protection for the breeders and also protection for this like companies, it got increased. So that's why from here onwards the majority it is being controlled or else it is being done by this private sector only, right? So as of now, actually that is one of the problem which we say, in a way it is problem, in a way it is also a solution. We say it is a problem because we always think that this private entities, they work for only profit and there is no like positive development and all, but essentially they are doing this service for which they are charging these prices. Of course at times it might be costly, but at the same time they are also ensuring the quality, correct? So that is what you should be looking. So next, after this 2015 so government itself is trying to find solution for this exploitative behavior of this companies. Now of course like everybody can understand, so like all the burden of the seed sector cannot be taken by the government itself. It has to be like balanced between both public and private. So for that, this RKVY is a project, that is Rashtriya Krishi Vikas Yojana. So in that we have an integrated approach for public-private development model itself. I think I have a slide for that. In the next slide I will show you. But essentially those sort of schemes which is promoting like public and private partnership in the seed sector, it is being promoted from this 2015 onwards. Clear? So I think, yeah. So this is that program. So if you can read a public-private partnership for integrated agriculture development, that is PPP IAD. So this is under Rashtriya Krishi Vikas Yojana program, right? So this is like, for example, what it is trying to do. PRS 10, it is like a variety developed by this IARI, that is Indian Agriculture Research Institute. So this has been given to these private people for licensing. So in other words, they have this access. Now they can like mass multiply it, and they can go for like commercial selling of it. So same thing, the varieties developed by this IVRI and then IAHR, IARI. So these were like shared with the private sector for commercialization without like through this non-exclusive licensing. So they have given this non-exclusive licensing. Now private companies can go for this marketing aspect. So that's why, see, under this few domains, that is under this production and marketing, the private entities have been given license to invest there, and they can also do this service, and also they can realize some profits. Correct? So this actually, when the question is like explain the role of like PPP, so first you can give that brief evolution. After that, when you are explaining the role, it is not just in the role of marketing. So you can start from like what is that production, seed production point of view. After that, ensuring this quality supply chain. After that, you can talk about this marketing, and also you can talk about like export opportunities. So if you explore all these areas, then there will be like comprehensive or as like broad coverage of themes in the answer. So that would be better, right? And in the end, also you can say for conclusion that depending on government, all the time it may increase the burden on the government, both fiscal burden and also like moral burden. So it is like duty of the citizens to share the burden and also do this welfare to the society. So on those lines, something you can make up for conclusion. Clear? Yes? Okay. So this is like important aspects about this seed science. Now we will look for this okay. Now we will understand this plant physiology. And again, as I told you, like these are like interlinked topics. So again, plant physiology, it is like one of the easiest topic to read and also like easiest to score also. So under this, I have like taken few of the basics plus like applied part, which could be like a potential question in this year mains. So first one is SPAC, that is soil plant atmospheric continuum. So this is again like very important topic, having interlinkages with this soil science also, right? In soil science, we read about like soil moisture constants. Especially we just focus mainly on this soil moisture aggregates, why there is like soil moisture crisis, all those things we'll be looking into. Here the science behind that, essentially water is in this continuous cycle. So hydrological cycle we say, but at the same time starting from like groundwater to this humidity, that is water vapor in the atmosphere, there is a continuous cycle or a continuous chain, and this chain is ensured via pressure difference. So what is that we are going to understand, but essentially that is like very important topic for this year, that is SPAC plus soil moisture constants, right? So that is one. Next one is plant growth regulators, and this is again like asked in number of times, but essentially this is one of the most important topics. And essentially here also there is no problem for like content. So adequate content is present both in like Tamil Nadu agro material and also like ICR material. Here the challenge is presentation, right? How you are going to present it. So what are the important points you are going to do? See here only one advice I can give you for PGR, that is plant growth regulators, is like prioritize your content. So and ensure that there is flow of points or is flow of thoughts in your points. That is to say, let's say for auxins only somewhere around 10 points you might be knowing. So but essentially if at all the question is like overall PGR, that is plant growth regulators, or else even if they have asked you to explain like 3 or 4 types of this PGR, then it is much better for you to like prioritize and write top 5 or 6 points from your 10 or 15 points rather than like dumping all the 10 or 15 points, right? Okay. So that is for PGR. Next one is like enzymes, and enzymes, it is going to be very important. So last time they had asked this enzyme lock and key model, right? But here enzymes plus factors affecting it, that could be one of the potential question for 10 markers. Clear? Next one is cyclic and non-cyclic photophosphorylation. See, essentially this cycles, that is C3, C4, CAM, so that has been asked adequately, but this photophosphorylation, though it is asked, it is like very less. But this year, it could be a potential question for 15 or 20 markers, right? So do like know that Z form of like, electron transport system that is happening, and how much energy is being produced and how much energy is being utilized. So those sort of things should be clear for you. Next one is mechanism of protein synthesis. So here the role of ribosomes, and then strand, mRNA, rRNA, tRNA, what is its role, that basic things you should be knowing. Next one is difference between photoperiodism and vernalization. So basically photoperiodism is like, altering the day and night, or else like light and dark conditions. And in this vernalization, it is basically like cold treatment to induce this flowering, correct? In both, the intention is to induce the flowering only. In one, we are like altering the light and dark conditions. In one, we are going for this cold conditions. Clear? Now we'll look for another few other topics. Sorry. We'll look into this in brief. So first we'll look into this, SPAC, that is soil plant atmospheric continuum. So in SPAC, if you can observe this diagram, see, first important concept you should be understanding from your geography point of view, and also from common sense point of view, is that this movement of water, it will happen from high pressure towards this low pressure. Yes or no? If you take this monsoons also, because India is going to have like extreme low pressure belt, especially during this peak of the summers, that is during monsoon. So that's why rain clouds will be moving towards this India, or towards this low pressure. Here also it is the same thing. So water moves from high pressure to low pressure, just remember that. Now if you take this soil system, the pressure at which this water is held by this soil particles, it will be somewhere around this minus 0.5 bars. And if you take this roots, and within this roots we have some pressure of like minus 4 bars. Now obviously there is a difference, correct, between this roots and this soil system. And also you would have heard about this Munch mass flow hypothesis, correct? So there they say that this diffusion pressure deficit, that is basically like because of this transpiration, evapotranspiration, a force is created in the plant, so that will help in sucking up this water plus this pressure. So combined together, so the water will be moving from soil towards this roots, and in the last year they asked the process, or else like phenomenons which is happening in this extraction, or else like absorption of like water. So basically imbibition, osmosis, diffusion. So these are the three main processes which is there, so through which it will be entering into the plant root. So once it is in the plant root, because of this pressure difference within this stem and this root, so in stem again it is like minus 7 bars. So now it will be moving towards this stem, and from leaf it will be transferred into this atmosphere in the form of evapotranspiration. Are you getting? So this is like a continuous cycle which is from groundwater towards this atmosphere, and it is being circulating. So that's why it is called as soil SPAC, that is soil plant atmospheric continuum. Clear? So next one is this thing, soil moisture constants. So basically available water which is available for the plant, it is called as capillary water. It is the water present between this field capacity and this permanent wilting point, correct? If you can see here in this hygroscopic water which is not available to the plants, so here the pressure at which this soil particles is holding this water or moisture is very high, so that the plant has to spend a lot of energy. That's why it is not possible for the plant to take up the moisture from this pressure. So that's why the available will be in this capillary region. So because of this, like two forces you would have heard, adhesive force and cohesive force, a continuous like column of water will be created. It is called as capillary rise, right? So because of this, the water will be moving towards this plant root zone, and it will be absorbed by the plants. So that's why this concept, that is SPAC, it is going to be important for your examination. Up to here it is clear, everyone? Right. Next one, the differences between this photoperiodism and vernalization. So if you want, you can take this screenshot also, right? Okay. So basically, as I told you, like photoperiodism is like altering this light and dark phases for these plants to induce this flowering, correct? So basically we have like three types of plants, that is long day plants, short day plants and day neutral plants. For example, tomato, again it is a day neutral plant. So day neutral plant means it is not having much effect on this altering light and dark phases. So it can like induce flower in the neutral conditions itself. So in vernalization it is basically like inducing these cold temperatures or as like cold conditions to induce the flowering in these plants. So that is called as vernalization. Clear? Right? And rest of the differences I have provided here. See, again it is like pretty like a simple concept, and hardly it might be asked for 10 or 15 marks. So this much content would be enough for you, right? Okay, next topic we will be looking is plant protection. Okay. See, in this plant protection, main important aspect, yesterday also I told you, what you have to understand is that basic aspects. For example, last year they had asked for this, EAL and ETL, correct? So in the first or second class I explained you, that is EAL is like economic injury level, economic threshold level and general equilibrium point. So that is the basic concept to like define or else to understand when insect is going to become pest, because all insects are not pest, right? So that is why that knowledge is important. So here like these are the expected areas for this year mains. So because the prices of tomato was skyrocketed, again like 200 rupees, 150 rupees, that was the price. It was not because like usual thing happens, that is artificial scarcity, but this time actually the yield itself was less because of this disease, that is leaf curl and mosaic virus. So because of this there is like 50% yield reduction in the tomato plants, and that's why now there is inflation in this particular crop. So that's why that question could be asked. But essentially, if at all that question is coming, not just like generalistic management practices, but essentially I have to give this IDM, integrated disease management procedure for this two diseases. Okay, let's discuss that in brief here itself. Right? So okay, so basically this leaf curl, so that is transmitted by, any idea? It is by whitefly, yes or no? It is transmitted by whitefly if you take mosaic virus, and again it is transmitted by aphid, correct? So these two differences you should be knowing, correct? Basically both are what? Sucking pest. If at all a sucking pest is there, the chemical control is systematic this thing, pesticide. So one of the example for systematic pesticide is imidacloprid. So here see, what you have to do is that you have to understand like types of pesticides and just try to remember at least 2 or 3 examples with their active ingredients. So that much is good enough for you. Clear? So that is for this leaf curl virus and mosaic virus. And in fact like both viruses belong to this Gemini viridae, correct? So both belongs to this Gemini viridae. Brief biological understanding of these viruses and then symptoms of it and then management procedures, that is what you should be needing for this leaf curl and this mosaic virus, right? So next one is major storage pest. So this is like one of the most important and repeated question, and it is pretty easy, like hardly we have like 10 to 12 important storage pests. So first know the little bit biology of them, and then the major symptoms it will be causing and the like curative measures, right, and also preventive measures. So next one is like in the syllabus actually we have something called as like plant quarantine measures, correct? So one of the like disease which is listed in this plant quarantine diseases is BBTV, that is banana bunchy top virus. Because this is not native to India, this is like it came to India via this seed materials, because one of the main source for this banana bunchy top virus, I think I have a slide for that, yeah. So if you can see here the entire bunch or as the leaves, it will start to like cluster together, it looks like kind of rosette formation, correct? Here see, the important thing is that when you are defining this sort of symptoms, utilize proper terms like rosette formation, cluster, right? So this sort of like words should be used here. So there will be less production of the fruits, and in many cases there will be no production of the fruits, correct? And so major thing is that it is transmitted from this seed material from one place to another place, and also it is at times it is transmitted from this aphids, right, at times, but most of the times it is like mechanical transfer of this disease from this either farm equipments or else from the seed sources. Clear? So that is banana bunchy top virus. So here again you should be knowing little bit about this plant quarantine measures which is present in India. So that is plant quarantine measures. Essentially, if you take this, international conventions are also in place, and also like domestic measures is there. So do have a brief idea about those things. Next one is IPM and IDM. This could be asked for 15 or else like 20 marks. So in IPM, yeah. So this slide it will give you a brief idea. So but the thing is that, see, when you are explaining this IPM, so biological, mechanical and chemical and cultural, so this will be there. So along with that, so do try to like incorporate this management aspects. That is to say weather forecasting, that is one important thing, and then planning according to the thresholds, that is EAL, ETL, that is one. So do incorporate this sort of diagram in the answer, so that would be much more comprehensive and holistic rather than just these three or as these 4 points. So in the IDM, that is integrated disease management, the major aspect you should be knowing is, see, for example, host plant resistance, that is the important source. So that start from there, then from field sanitization, that is removing this debris from the field, because that debris itself is like one of the potential source for this pathogen to survive, right? So remove those pathogenic survivals and then go for this tillage and fertilizer application. There itself we can manage most of the things. And in the last case, we can go for this chemical and biological methods for removal or as like management of this pest and diseases. Clear? Yeah. And last one is this biocontrol of pest. See, in this biocontrol of pest, one thing you should be understanding is, see, obviously there is one way of like biological control of pest, that is increasing the population of natural enemies. So for that, one of the ways is that ensure that there is adequate population of natural enemies in the natural ecosystem. One. Second thing is that if at all that natural population is not good enough, then you have to mass multiply and you have to introduce that into the field conditions, right? So these are the two main ways in which we can increase this natural enemies population. So that you have to like little bit have the idea of these examples, that would be good enough, right? So next we will look for this is horticulture. And I told you like yesterday also, if you take paper 2, it is like genetics, breeding plus horticulture. So combine these three together, you will get more than 200 marks. That is out of 400, just 3 subjects, more than 50% marks is coming from these 3 subjects. So in this horticulture, especially from last 3-4 years, one of the major topic is this thing, protected cultivation. So under protected cultivation, we have many types. For example, corrugated is there, for example, shade net, for example, greenhouse gas, for example, low tunnel technique, and then we have plastic greenhouse gases. N number of types is there. At least understand major types of this protected cultivation. And in fact, you won't believe that this protected cultivation, it is present in this NCERT itself. So if you go back to that NCERT, read that document. Sorry. Okay, read the document, that will give you like adequate content for your protected cultivation question. So here the questions could be like this. So what is protected cultivation? What is the status of it in India? What is the potential of it? What are the types of it? Challenges, solutions and government policies. So these are the ways in which they are going to ask the question, right? And especially thus far the questions have been on, one is types, second one is on like potential and utilization of it, right? But this year the challenges and also like types could be asked. I think, yeah, so this is brief types. For example, high-tech polyhouse is there, shade net is there, poly houses is there, then polycarbonate house is there, glass house is there, and this Ladakhi structure. So with utilizing this mud form, we are going to create this greenhouse gas conditions, greenhouse conditions. So this is like types of like protected cultivation, and in number of crops, essentially like these are the few examples I have given, especially those which have like export potential and also which is like difficult to grow in the like field conditions. And if at all you are growing in the protected cultivation, then the quality and also quantity will be more. So those sort of crops will be grown. So for example, carnation, for example, lily, for example, this anthurium. So these are, tulips. So these are the ones which will be usually grown. And also like if you take this vegetables, this cherry tomato, the small one, which will be used in this salads. So those sort of things will be grown in this protected cultivation, right? So that is one of the theme area, main theme area. So next one is, okay, this one, status, challenges and solutions of cashew crops. So cashew is an example of which type of crop? It is a plantation crop, correct? So here, see, the question could be like this. So what is the potential, what are the different types of like plantation crops grown in India? That could be one question. Or else they can ask this cashew itself as a separate question. Here you should be knowing the status of it, where it is being cultivated, what type of soil is needed, and then what is the few of the varieties names, and then you can think of this, challenges and solutions. So for the cultivation of cashew, right? And usually cashew is what? Semi-arid agriculture crop, definitely irrigation is a problem, fertilizer management is a problem, pest and disease is a problem, right? So the quality is not being assured, first thing. Second thing is that, this cashew apple, so that itself is one of the like rich source of this vitamin C. So proper utilization of that, and also like there is this, alcoholic product prepared from that, it is called as fenny, right? So this sort of like value addition is also needed in the crop. So those are the things you can bring in this cashew cultivation. So next one is like POP of chrysanthemum. So thus far what has happened is that especially POP on this flower crops. So that has been like focused less, and also another crop is like medicinal and aromatic crops. So that's why the focus on these two crops have been less, but I believe that at least like one of the important crop, if I say like aromatic crops, I can medicinal and aromatic, I can say sarpagandha. One time it has been asked, or it's like aloe vera, again only one time it has been asked. So at least for these crops, a generalistic POP. So usually in these crops, they won't be asking like specifics. I told you like in rice, wheat and maize, they will be going for specifics. But here, usually they will be asking like holistic management, holistic POP. So just have that brief idea about this, one is like sarpagandha, aloe vera, for this medicinal and aromatic crops, that is good enough. Clear? So protected cultivation, just now we discussed. Then post-harvest technology. So here, let's say for example, making of this jams, jellies, marmalades, that is one way. Second way is that holistically asking, what is the like loss, or is like, economic loss which is occurring because of this post-harvest losses, and also what are the ways in which we can increase this or else reduce this post-harvest losses. So these are the major theme areas for this post-harvest technology. And last one is principles of landscaping. And again, this landscaping and gardening, the questions have been less. But if at all they are going to ask, usually they will be asking for 10 markers. And rare cases, they may be asking for 15 markers. So here the principles of landscaping, minimum that much knowledge should be known to you. Right, I think I have a, So these are the major 6 principles of this landscaping. So one is like unity, and then this sequence, symmetry. So these are the major features of this, landscaping. So understand these principles, explain them in brief with examples, that would be good enough. And last important subject in our syllabus is food science and nutrition. So in this food science and nutrition, and again like, see here it is actually one of the easiest subject to score, because the syllabus which are being given here, it is like almost same as like GS3 portion which we have. Only additional things are like, vitamins, minerals, their role, their deficiencies, and what is the role of like, balanced diet, and what is this dietary recommendations. So these are the like extra topics present in the syllabus, but as a whole, it is like most of this is in our GS3 itself, right? And also few aspects of like governance will also be covering this, right? So these are the expected areas for this year mains. One is ways to strengthen this food security in India. See, first thing, if you look at the syllabus, first heading itself is like, food production and consumption trends in India. So last year that was asked, like 50 years trend was asked. But if you like even know this generalistic idea, so India is one of the like largest producer and exporter of many like agricultural commodities. But still, if you take like global hunger index, where India is ranked 107 out of like 122 nations, and also let's say for example if you take malnutrition, if you take hidden hunger, so and also if you take like child wasting, stunting, so in every of this aspect India is lagging behind. So if you have like a luxury of production, obviously there should be good health conditions, but that is not happening. So that's why this question will assume significance. So that is ways to strengthen this food security of India, correct? So Sorry. Next one is hidden hunger and PEM. So hidden hunger is like micronutrient deficiency. So usually focus will be provided for this, carbohydrates and sometimes of course pulses, that is basically protein and fat. But essentially this micronutrients is not given that much of significance. So that's why because of this we have a condition called as hidden hunger. So outside, just by looking at a person, he might be looking healthy, but essentially the physiology of that person is not healthy, correct? So that is hidden hunger. So reasons for that and ways to overcome those things is important. Next one is PEM, that is protein energy metabolism. So here, see what is happening because of low consumption of protein. So the body will develop certain disorders. For example, you would have heard about this Kwashiorkor disease, right? So where bone deformations, that sort of diseases are called as like PEM, protein energy malnutrition. So this is going to be a potential question for this year, right? Next one is national food security. India, especially after this NFSA, that is National Food Security Act, it is focusing more and more of ensuring both food security and nutritional security. Here it should be understanding what are the features of this national food security policy and what are the challenges of it, and you can suggest some solutions for it. And last important part is role of balanced diet for like healthy life. And especially these sort of questions, they may ask like role of balanced diet in like later stages of the life, that is one way of asking, or else balanced diet and child development, that is one way of asking, or else this balanced diet and adult. Basically depending on the life stages they may ask the question, or else holistically what is the role of like balanced diet in maintaining healthy life. So this also could be asked. Clear? Yes. Yeah. So then that is it. So these are the major theme areas and potential areas for like upcoming mains. And see, this initiative which we had launched in this insights, it is basically for like benefit of this students, because see when there is mains and the student doesn't have like clarity of what to expect from the examination, it would be tough. So considering this, we have started this free initiative. So of course like we will be conducting this full length classes itself, and I told you like who all can take this agriculture as optional and all. So you do watch these 4 videos, it will be giving you much more clarity. And if you like have that gut feeling, okay, this person is good enough, I can trust with this optional and all, so definitely you are most welcome. And I've been giving, I had given you my Telegram ID also. And for those who doesn't know, this is the Telegram ID. So KRTP12, so you can reach me at this Telegram ID. So if you have any doubt, you can ask me, right? And all the best for like upcoming examination for people who are aspiring and writing this year mains, and for those people who are aspiring for next year attempts, for them also all the best, and God may bless you with all the endeavors which you are like planning in your life, right? Thank you all. So if you have any doubt, of course, like please reach me at this Telegram ID, right? Thank you.